Okay, so these next set of slides, we're going to be walking through the motility of the mouth, of the esophagus, and then we'll show some brief images at the end of disorders of them. So this is an example of a local motor event that's restricted to the mouth and the esophagus. So the mouth is the first point of entry for food, and upon taking in food, you're going to start chewing it, or also called mastication. And there are several functions to this chewing mastication. You're going to start lubricating the food by mixing it with the salivary mucus, and this begins the first part of digestion because the saliva in your mouth contains enzymes such as salivary alpha amylase that acts on carbohydrates and breaks them down into smaller carbohydrate molecules. The mechanical breakdown of the food is also for better mixing in the stomach. It increases the surface area and makes it more manageable for the stomach. And this whole process is generally voluntary, although there tends to be a sort of a reflex aspect to it. If you're sat in your chair and you have some gum in your mouth, you'll kind of unconsciously chew as an almost reflex. But you have complete control over this if you were to concentrate on it. The food then is passed down the esophagus, which is a tubular conduit about 20 centimeters long, and this is for food transport from the mouth to the stomach. There are a couple of structural aspects you need to sort of focus on. The upper third of the esophagus, the circular and longitudinal muscle layers, are striated like skeletal muscle, and then as you go down, you get a coexistence of both striated muscle and smooth muscle that is consistent with the rest of the GI tract. And then the lower third is just purely smooth muscle. So the neuronal control of the esophagus is shown in this slide, and there is both afferent and efferent innervation. The afferent innervation sends feedback to the swallowing center, and this is how a peristaltic wave starts during swallowing. The signals get sent back to the swallowing center and regulated, as we see in a couple of minutes. The efferent is mainly by the vagus nerve, and there are several points to take note of here. The vagal somatic neurons are to the striated muscle at the top third of the esophagus. Then you have vagal visceral motor neurons that are preganglionic and parasympathetic, and these go to enteric nervous system sites through the myenteric plexus. And then finally, these myenteric plexuses send out nerve branches to the smooth muscle, and these are generally cholinergic muscarinic synapses that are atropine sensitive. There are two sphincters in the esophagus: the upper esophageal sphincter that prevents entry of air into the gastrointestinal tract, and the lower esophageal sphincter, which is a zone of elevated pressure at the bottom of the esophagus. It's very important for preventing acid reflux into the esophagus. A disorder of this sphincter is very common, and as far as gastrointestinal diseases, is one of the main reasons a person would go to their practitioner for help. So the contraction of the sphincters is by cholinergic nerves, but these are generally nicotinic and therefore atropine insensitive. And there are sympathetic nerves that run to it as well that are adrenergic. The relaxation, which occurs during peristalsis, is an inhibitory nerve signal sent by the vagal nerve to the circular muscle of the lower esophageal sphincter. The transmitters that are released to relax this are vaso interactive peptide and nitric oxide, and there's also a reduction of vagal excitatory fibers through the nicotinic cholinergic receptors. I want to point out here that these nicotinic receptors in the sphincters are exceptions to the general rule that most of the cholinergic receptors in the GI tract are muscarinic. And so we can see here at the bottom this graph of how relaxation and contraction occurs in the sphincters. You have vagal inhibitory fibers on the top and vagal excitatory fibers on the bottom, and under normal conditions the sphincter is contracted and there's sort of basal excitation that maintains this rate, and then during the act of passing food down the esophagus, this will relax, and what happens is the vagal inhibitory fibers increase their firing rate, and the excitatory fibers then reduce that, and you get this relaxation that occurs. And then to close it again, the vagal excitatory fibers increase their firing, and this in conjunction with reduction of firing of the vagal inhibitory fibers then promotes closing. So just to reiterate something you've seen in previous slides from earlier lectures, the sympathetic and parasympathetic innervation of the gastrointestinal tract is shown here in general strokes. You have sympathetic, which are short preganglionic fibers that are cholinergic in the preganglionic synapse, and then they have long postganglionic fibers that generally end with the release of norepinephrine onto adrenergic receptors. The parasympathetic has long preganglionic fibers, and both pre and post ganglionic synapses release acetylcholine. And you can see on the sides here that the preganglionic are generally nicotinic and the postganglionic cholinergic are muscarinic, but as we saw on the previous slide, there are exceptions such as the sphincters. So swallowing is initiated by voluntary control, but then as soon as the bolus of food begins its passageway down, the swallowing reflex is enacted. And this is a sequence of events that pushes the food bolus from the mouth down into the stomach through the esophagus. And there's three phases: the oral voluntary phase, the pharyngeal phase, and the esophageal phase. So this is a section of figures showing the side view through the body, so we can see how the food bolus is moved during the act of swallowing. So after chewing and mastication, you create a food bolus, and then this is pressed against the top of the mouth by the tongue and pushed backwards, and then the sensory nerves at the back of the throat then sense that the food is there, and this initiates the swallowing reflex, followed by a peristaltic wave. So the food is squeezed and pushed further down into the throat, and then the epiglottis folds down to close off the trachea, and then the esophagus relaxes and the food bolus is pushed down into the esophagus. So the control of esophageal motility is through the sensory receptors in the mouth and the pharynx, passes the signals to the swallowing center in the medulla, and then these through vagal nuclei and non-vagal nuclei passes the stimulation, the efferents to the somatic fibers and the striated muscle of the esophagus, and then the visceral motor fibers in the myenteric plexus that then control the smooth muscle contraction and relaxation of the peristaltic wave. This is a pressure profile during the pressure wave that goes down the esophagus during swallowing. and the different pressures at the different parts of the esophagus are shown here on the right. And so at rest you can see there's a certain amount of pressure in the esophagus itself. You can see that at the sphincter levels that the ambient pressure is higher than in the rest of the esophagus, and these are because the constriction of the muscles of the sphincters increase the pressure at the points obstructing flow or regulating flow. And so after swallowing, we then see this pressure wave move above the pharynx. There's a brief increase in pressure as the food bolus is pressed towards the back, and then the upper esophagus sphincter will relax and then recontract as it allows food to pass, and then this pressure wave of peristalsis follows, pushing the food bolus down into the stomach. And we can see at the bottom the lower esophageal sphincter will actually relax before the food bolus actually gets near to the stomach to allow for ease of access into the stomach, and then after the food passes it constricts again, similar to the upper sphincter. It's important to note that the thoracic cavity is generally held at a negative pressure, and this is because the chest wall generally pulls outwards and the lungs pull inwards, and so there's a negative pressure there. And the esophagus is just a sort of soft tissue. It's quite malleable, and so the insides of the esophagus, even though these numbers look at zero, it's actually negative pressure in there because of the negative pressure in the thoracic cavity. This pressure difference is passed into the lumen of the esophagus. This is another diagram showing the pressures at rest of the different parts of the esophagus, and this is in millimeters of mercury. So it's ambient pressure or atmospheric pressure in the mouth, and then the upper esophageal sphincter is a high pressure area, and then the lumen of the esophagus is actually negative, as pointed out in the last slide. And then we come to the lower esophageal sphincter, which is another high pressure point. And then these diagrams reiterate how the pressure wave flows down through the esophagus. As well as the thoracic pressure affecting the esophageal luminal pressure, breathing also affects it, as shown in the red box here. The pleural pressure starts at about -5 centimeters of water, and with inspiration that will decrease, and then with expiration it will come back down to the normal level. And this just shows that the intraesophageal pressure of the lumen is determined by the thoracic pressure and the changes to that due to breathing. If you were to eat a lot of food and take a mighty swallow, and the initial pressure wave of peristalsis pushing that food down isn't enough to clear the esophagus, then you'll get a second peristaltic wave occurring. And this is an example of an enteric nervous system reflex that's just intrinsic. And what I mean by that is that everything that is required for this reflex is found within the GI tract itself. And so the sensory receptors in the esophagus will sense that there's still food in there, and that will send signals to the myenteric plexus, and that will initiate a second peristaltic wave that pushes the food through the esophagus. This is an example of an enteric reflex, like I said, and you will see in the literature what's described as the law of intestine, and this is essentially a great example of that. The fact that the whole of the GI tract can sense and respond to luminal contents based purely on intrinsic nerve functions, and this figure here shows in general terms how that happens. So if you have food left in the esophagus, around by enterochromaffin cells, then whether it's chemical, mechanical stimulation, these cells will release serotonin into into the organ, and this will be picked up by sensory neurons, and these then will put neurotransmitters. The neurotransmitter in this case is calcitonin gene-related peptide that will then activate the nerves in both the myenteric plexus and the submucosal plexus, and there's a characteristic fashion of this. Basically, distal to the food, i.e. closer to the anal side of the GI tract, you get relaxation caused by nitric oxide and vasoactive intestinal peptide, and this will cause the smooth muscle downstream from the food bolus to relax, and on the distal side closer to the mouth you get release of acetylcholine and substance P, and that causes it to contract, and so you get relaxation distal, contraction proximal to it, and this pushes then the food further down in an arboreal direction, in the orthograde from mouth to anus. So the GI tract is a very integrated system, and we're just going to talk a few minutes about the neuronal control of GI function. We talked briefly then about enteric reflexes, and they play a major role in controlling the motility, secretory and absorption functions of the GI tract. And these enteric reflexes are intrinsic with the law of intestine that we just talked about, and the elements of the reflex are located completely within the intestinal wall. And then there are long-range reflexes that are extrinsic autonomic pathways between the guts and the CNS that are involved. This is a diagram that explains that in more detail. So you have mechanoreceptors, chemoreceptors, temperature receptors, and pain receptors that can send signals both to the central nervous system or just to the local afferents, the myenteric plexus and the submucosal plexus. If they just go through the myenteric and submucosal plexus, this is local, and this is part of the enteric nervous system. And then if they use splanchnic or vagal afferents into the central nervous to control the muscular interna or endocrine cells, then that's a long range because it involves both the enteric and the central nervous system. This is a more detailed figure of how the long-range signals can affect the enteric nervous system. We have parasympathetic on the left here. The vagal and the sacral cord affect the enteric nervous system directly and alters its function, and then the enteric nervous system will go on and affect smooth muscle secretory cells, endocrine cells, and blood vessels. And so this is an indirect way that the parasympathetic controls the enteric nervous system functions. Sympathetic is similar in that you have postganglionic fibers that affect the enteric nervous system. The one exception here is that the sympathetic ganglia affect the blood vessels of the GI tract directly, and so they kind of have this short circuit that they don't have to go through the enteric nervous system to regulate blood supply. And so the control of esophageal motility is both through long range and short range. The swallowing is a long range reflex that uses the swallowing center, and then the secondary peristaltic waves is a short range reflex It is intrinsic to the GI tract. Okay, so I'd like to finish off this short lecture by talking about some disorders of the esophagus pertaining to its motility. And I think they kind of illustrate where things can go wrong, and it rounds out your understanding of just what the esophagus does. So achalasia is a neurogenic disorder with impairment of the esophageal peristalsis, and the LES contracts and then doesn't relax properly, and this causes a high pressure blockage that the food bolus is unable to get through, and you get food stuck in the esophagus, and that can lead to dissension and problems with peristalsis, pain, etc. You have diffuse esophageal spasms where the coordination of the contractions are out of sync. And you get reflux because of this, and it's painful, and the esophagus often has a kind of a corrugated look to it. Sclerodermia is where fibrotic tissue just is found everywhere because the smooth muscle becomes fibrous, and that just prevents contractions from happening altogether. The most common of all these is gastroesophageal reflux disease. I mean, everybody feels it at some time, but some people suffer from it chronically to the point of damage to the lower esophagus and the esophageal sphincter, which then perpetuates. And then you have the hiatus hernia, where the opening through the diaphragm that the esophagus goes through is widened for some reason, and then the stomach protrudes through that and pushes the esophagus up, and you can have pressure on the heart as a result, and quite severe pain due to it. So the endoscope is a common tool that's used to examine and differentiate between the diseases. It can be fed in through the nose or orally, and can go down even further into the small intestine, and it can be used to visualize the issues. It can be used for biopsies, and it can also be used as a pressure sensor. So these are some endoscopy pictures pertaining to GERD. You have the normal at the top, and then erythema after a while with the reddening and inflammation due to acidic damage, and then with persistent exposure you get isolated erosions followed by confluent erosions, and then circumferential erosions, and eventually you get strictures. And this is not only finds it difficult to contract but difficult to relax. It's just a fibrous valve that is very painful and really doesn't do what the esophagus sphincter is supposed to, which is constrict and relax at certain opportunities. So this is a hiatus hernia X-ray. You see the diaphragm here between the light and the dark, and then this wide gap in which the stomach is protruding through, and you have the esophagus up here. This is a normal one. Here's the stomach, and the esophagus is pointing upwards. And